Welcome to What's New, your channel for everyday solutions.
Be sure to subscribe, share and like our videos. This is What's New.
Stem cells edited to fight arthritis. Goal is vaccine that targets inflammation in joints. By Jim Dryden. Using CRISPR technology, a team of researchers rewired stem cells' genetic circuits to produce an anti-inflammatory arthritis drug when the cells encounter inflammation. The technique eventually could act as a vaccine for arthritis and other chronic conditions. Using new gene editing technology, researchers have rewired mouse stem cells to fight inflammation caused by arthritis and other chronic conditions. Such stem cells, known as smart cell stem cells modified for autonomous regenerative therapy, develop into cartilage cells that produce a biologic anti-inflammatory drug that, ideally, will replace arthritic cartilage and simultaneously protect joints and other tissues from damage that occurs with chronic inflammation. The cells were developed at Washington University School of Medicine in St. Louis and Shriners Hospitals for Children St. Louis, in collaboration with investigators at Duke University and Cytex Therapeutics Incorporated, both in Durham, North Carolina. The researchers initially worked with skin cells taken from the tails of mice and converted those cells into stem cells. Then, using the gene editing tool CRISPR in cells grown in culture, they removed a key gene in the inflammatory process and replaced it with a gene that releases a biologic drug that combats inflammation. Our goal is to package the rewired stem cells as a vaccine for arthritis, which would deliver an anti-inflammatory drug to an arthritic joint but only when it is needed, said Farshid Gillick, Ph.D., the paper's senior author and a professor of orthopedic surgery at Washington University School of Medicine. To do this, we needed to create a smart cell. Many current drugs used to treat arthritis, including Enbrel, Humira and Remicade, attack an inflammation-promoting molecule called tumor necrosis factor alpha, TNF-alpha. But the problem with these drugs is that they are given systemically rather than targeted to joints. As a result, they interfere with the immune system throughout the body and can make patients susceptible to side effects such as infections. We want to use our gene editing technology as a way to deliver targeted therapy in response to localized inflammation in a joint, as opposed to current drug therapies that can interfere with the inflammatory response through the entire body, said Golick, also a professor of developmental biology and of biomedical engineering and co-director of Washington University's Center of Regenerative Medicine. If this strategy proves to be successful, the engineered cells only would block inflammation when inflammatory signals are released, such as during an arthritic flare in that joint. As part of the study, Gillick and his colleagues grew mouse stem cells in a test tube and then used CRISPR technology to replace a critical mediator of inflammation with a TNF-alpha inhibitor. Exploiting tools from synthetic biology, we found we could recode the program that stem cells use to orchestrate their response to inflammation, said Jonathan Brunger, Ph.D., the paper's first author and a postdoctoral fellow in cellular and molecular pharmacology at the University of California, San Francisco. Over the course of a few days, the team directed the modified stem cells to grow into cartilage cells and produce cartilage tissue. Further experiments by the team showed that the engineered cartilage was protected from inflammation. We hijacked an inflammatory pathway to create cells that produced a protective drug, Brunger said. The researchers also encoded the stem, cartilage cells with genes that made the cells light up when responding to inflammation, so the scientists easily could determine when the cells were responding. Recently, Gillick's team has begun testing the engineered stem cells in mouse models of rheumatoid arthritis and other inflammatory diseases. If the work can be replicated in animals and then developed into a clinical therapy, the engineered cells or cartilage grown from stem cells would respond to inflammation by releasing a biologic drug, the TNF-alpha inhibitor that would protect the synthetic cartilage cells that Gillick's team created and the natural cartilage cells in specific joints. When these cells see TNF-alpha, they rapidly activate a therapy that reduces inflammation, Gillick explained. We believe this strategy also may work for other systems that depend on a feedback loop. In diabetes, for example, it's possible we could make stem cells that would sense glucose and turn on insulin in response. We are using pluripotent stem cells, so we can make them into any cell type, and with CRISPR, we can remove or insert genes that have the potential to treat many types of disorders. With an eye toward further applications of this approach, Brunger added, the ability to build living tissues from smart stem cells that precisely respond to their environment opens up exciting possibilities for investigation in regenerative medicine.
Welcome to What's New, your channel for everyday solutions.
Be sure to subscribe, share and like our videos. This is What's New.